By the middle of the 19th century, scientists understood that traits could be inherited from parent to offspring. So they knew that something was responsible for inheritance, that it didn't just come out of thin air. The chemical components that make up what we now know of as DNA, which is responsible for inheritance, these components are too small to be seen. Okay? But with experiments and imaging techniques that came along in the middle of the 19th century, these have allowed for scientists to infer the structure and also the components of DNA. DNA is a polymer that's a natural polymer. So there are artificial polymers like plastics, but natural polymers like cellulose and starch, the carbohydrates, which are polymers of glucose monomers. DNA is a polymer made out of nucleotide monomers. The individual nucleotide monomer has three main components. So those components are a sugar ring, also a base, and a phosphate. Now the bases are the only part that are different on a nucleotide monomer. Similar to amino acids, that amino acid monomers are all the same except for just the side chain. There are 4 different nucleotide monomers, and the only difference between the 4 different nucleotide monomers are the 4 different bases. The bases will also look at the detailed structure, but for now this is a cartoon. This is adenine, cytosine, guanine, and thymine. They're usually written as A, C, G, and T for their short names. What's shown in these cartoons is structures that fit together very specifically for the bases that pair. Adenine always connects to thymine, and cytosine always connects to guanine. This is what's known as complementary base pairing. The structure of DNA is a ladder that's twisted. So that's what's shown here. And the rungs of the ladder are these complementary base pairs. The sugars and the phosphates, that's the flat part and the red part. The sugar and the phosphate make up the outsides of the ladder, and the rungs of the ladder are the base pairs. These are the structures of those 4 bases: adenine, guanine, cytosine, and thymine. Nucleotide monomer of DNA, the only thing that's different is this one portion, which is the base. So the phosphate is something we've seen before. This is PO4 -3 charge. That's the polyatomic anion. This is a sugar ring. We've seen sugar rings before. It's a carbohydrate. Some of the oxygens have been removed because for a carbohydrate, every carbon should have an OH and an H, but this one has one less OH group here. So this is a ring, and the phosphate and the sugar make up a DNA backbone when we put this together. The bases do change for each monomer, and that's what actually makes up the DNA code. When you put these parts together, this is an individual nucleotide. It has a sugar ring, and then on one side of the ring is a phosphate group, and on the other side across from it is a base. And this will become important, the locations, the fact that this base is always on one side of the sugar ring and the phosphate group is always on the other side. Well, when we put these nucleotides together, they always have the bases on one side. So it's covalent bonds that connect these 3 components. This is a covalent bond here, and this is a covalent bond here. And it's also covalent bonds that connect the monomers. One nucleotide is connected to the next nucleotide monomer with a covalent bond. Here's a representation putting together 3 nucleotide monomers. This is the yellow, blue, and pink. This is a single nucleotide monomer. And this is another yellow, blue, and pink. This is a second nucleotide monomer, and then this is the third nucleotide monomer. You can see in this representation that the bases are always on one side. They're always on the right side, directly across from phosphate, both connected to the sugar ring. So what connects the individual monomers together is the sugar ring of one is connected to the phosphate of the second one. Okay, and then the sugar ring of the second one is connected to the phosphate of the third one. And so this sugar ring of base number 3 should be connected to the phosphate of base number 4. And so what you have is this yellow and blue portion is called the backbone. What connects the monomers in the DNA polymer is the phosphates and sugars. What hangs off the side is all of those bases, so the A, C, T, and G. And that allows for those bases to pair up in the three-dimensional shape. Here's a single DNA strand shown with all of the atoms. So you have the yellow and blue. This is the phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar for 4 monomers. So this is 4 nucleotides here. The 4 nucleotides are different for the bases. So the bases hanging off the side are a thymine, an adenine, a cytosine, and a guanine. Here's a closer look at those 4 DNA bases. They're actually kind of similar. So each of these bases contains at least 1 nitrogen atom, or at least 1 amine group. This is cytosine, that's C, and thymine, that's T. So both of these have a ring, and there's 3 nitrogens on the cytosine, and there's 2 nitrogens on the thymine. This is a single ring. Adenine and guanine are similar in that they are 2 ring structures. So this is a 6-member ring connected to a 5-member ring. Same for guanine. Adenine and guanine have similar structures, and cytosine and thymine have similar structures. However, the shapes of adenine and thymine are the 2 that match as far as puzzle pieces, and the shapes of cytosine and guanine are the 2 that match as far as puzzle pieces that fit together. And the puzzle pieces fit together by the shapes and also are held together with hydrogen bonding.
These bases can form hydrogen bonds, and a hydrogen bond is from the partial positive hydrogen that's on one molecule to a partial negative oxygen on another molecule, or it can be a partial positive hydrogen to a partial negative nitrogen on another molecule. So the potential nitrogen and hydrogens that have the partial negative are in polar bonds. So that would include this nitrogen, or this nitrogen, or this nitrogen, this nitrogen, or this nitrogen, or this nitrogen, this nitrogen, this nitrogen, this nitrogen, this nitrogen, or this one, this one, this one, this one, this one. All those nitrogens have potential for hydrogen bonding. The oxygens also have potential for hydrogen bonding. So these oxygens are all in polar bonds as well. Now that would be hydrogen bonded to any of these hydrogens that are also in the polar bonds. So the hydrogen would have a partial positive. Not every single hydrogen, but just the hydrogens that are in the polar bonds. So that would be, this would be the possibility of hydrogens that are in polar bonds but in a different molecule. The dotted or the dashed line, the hydrogen bond. So the hydrogen bond is between the hydrogen of one molecule, and it is an attraction. It shouldn't be called a bond, remember that from chapter 8. It's only an attraction to a partial negative on an oxygen or nitrogen in another molecule. Because of the nature of these bases on the DNA backbone, not all of these that are circled are possible for hydrogen bonding. So this depends on exactly how these bases are bonded on those nucleotides. But this is what just showing the potential on each of these structures given here.